Hey everybody, we're going to take a look at chapter 14. Now we're doing chapter 14 in two parts, so I'm going to have you do some reading out of the textbook, and if you look online there are some questions there as well as in your course pack that'll have you look at one section of chapter 14, mostly the first part of that, and there's a lot to cover there, and I think a lot of it's important, and so I think it's better to have you read through that. Now of course I'd be happy to answer any questions there, but I try and make those questions in the review section pretty straightforward so that you can kind of just focus on some of the basic concepts of the immune system. So with the first part of chapter 14, you're looking at a lot of things that are based on anatomy and physiology, some of the physical barriers to infection. We'll kind of go over that here in just a second in a little more detail. And then towards the middle, you're looking at some of the ways that these body systems connect to each other, how the bloodstream and the lymphatic system overlap, for example. And then towards the end, which is what we're going to look at here in more detail, we're looking at some of the different cell types and some of the different types of immune responses and how they work on a more in-depth level. So kind of do it in two parts, and the textbook is going to cover some of the basics there. Okay, so if you haven't read that already, I suggest that you do that first, although it's not 100% required either way, but ideally you'd probably want to read the textbook questions first, answer the textbook questions, that is, by reading, and then come take a look at the video. So think about doing that if you haven't. Okay, so when we're looking at chapter 14 here, we're taking a look at some of the, as I titled it here, some of the details of innate immunity. Okay, so this is building on that foundation in the textbook here. So first of all, before I get into the PowerPoints here, I want to outline just a few things here. So when we look at our immune system, it's really a three-tier system, or what we call the 3 levels of defense. Okay, so 3 levels of defense, and I'm going to put that in quotes, partly because when I say defense, that's kind of a broad thing. Also because it's described differently in different textbooks, different sources, different textbooks, different individuals will sometimes describe these a little bit differently. So the first part, the first level, so to speak, is actually not part of the immune system, and this is why we call them the levels of defense as opposed to the 3 levels of the immune system, because really the defense itself as a whole partly involves the immune system, but it also involves what we refer to as barriers to infection. This is actually not technically part of the immune system. It's actually part of the anatomy and the physiology of the body. The way the body has been kind of constructed to prevent microbes from getting in, and really trying to at the very least, keeping the threshold of microbes down. Recall from chapter 13, we talked about the infectious dose, the minimum number of microbes needed to establish and cause infection. And really, one of the ways that the body reduces that is by simply blocking them from actually getting in and from attaching and from establishing at that minimum level that's often required. So barriers to infection. I always like to put a little side note below each one of these, and I like to just refer to this as AMP, anatomy and physiology, as it has to do with the natural, as I mentioned, anatomical and physiological structures your body uses. Again, not technically not part of the immune system. Okay, so speaking technically, this is not actually part of that system. And again, the book goes over this in more detail. We really won't touch on that here in the PowerPoint. It's really going to be something you read about. Now, the second level of defense does start into the immune system. This is technically where the immune system begins. And the second level is often referred to as the non-specific immune response. And it's also referred to as the innate immune response, innate immunity, I should say. That's how I have it titled on the PowerPoint. I'm giving it to you in both ways because you'll hear it described in both ways. So it's sometimes referred to as non-specific, sometimes referred to as innate. In general, the best way to think about this level of the immune system is that the body is responding to typically foreign microbes, although you get into allergies and all sorts of weird kind of ancillary reactions, but typically to a foreign microbe, either a virus or bacteria or fungi, protozoa. Those are the sometimes helminths, so 10 of 5 typical things there. And the way it typically works at this level is that the body is responding from a quote-unquote self versus non-self basis. And this is largely based on protein receptors on every cell. Every cell has a series of proteins on the outside of it. Let me just show this here. So here's a cell. And every cell has a variety of little proteins that stick up out of the membrane. And they have a complex three-dimensional shape. And if you were to zoom in and look at an actual cell and look at these in great detail, you'd find that they have really elaborate three-dimensional shapes. And the totality of these are the equivalent to like a facial feature. So the way that these look can be kind of thought of as like a face and all the different subtle characteristics of a face. And the way the immune system works is that it often determines what is part of the body versus what is not part of the body. So again, self versus non-self, determines that to a certain degree based on the nature of these receptors. So for example, your body, just do a real basic drawing here, your body has cells that will have very specific receptors that are identical to your own body. So they might look like a kind of a triangle shape, something like that here. Okay, obviously, to clarify, they don't actually look like that at all, but that's something that would be, you would have a unique shape to it. So that might be what your body's receptors look like. And then if we were to look at a foreign invading microbe, it might have receptors that maybe have a very different shape. Perhaps they look more like this, whatever shape that is, I don't even know. Okay, and so the idea is that each different cell will have a slightly different receptor, or a series of them really. They're really not actually a receptor, they're actually what we call a cell identifying marker, or an ID tag. But every cell has their own unique one, and the body will determine its own from that of foreign or non-self entities. And in this general sense, then, the body can kind of differentiate itself from other microbes. Now, it does this in a very kind of discriminatory way. It looks at its own receptors and determines that they are of the body, and anything else that doesn't match those to a certain degree can be considered foreign. Now, there's more to it than that, so I'm generalizing quite a bit here. But in a sense, that's kind of how it works. You either have the matching or you don't, and therefore you're either part of the self or the non-self. And so we'll talk about it a little bit more. These are an example, give you a heads up here of what is often considered non-self or referred to as PAMPs. And these are facial or cell characteristics that are found in certain types of pathogens. So they're often a very immediate type of receptor there, or receptor rather, that triggers an immediate response by the immune system. Okay, so that's the second level. Let me give you an example of that, and then we'll move on past this here. An example of what often carries out this self versus non-self determination, or something that we've talked about already, and these are phagocytes. Phagocytes are the cells that engulf and swallow foreign particles and foreign microbes, and they're often doing that based on a self versus non-self basis. So they travel throughout the body. If something looks foreign, they swallow it and they kill it, and if not, they leave it alone. There's other aspects to that. That is just an example, not the entirety of the second line. Just one example. Okay, now the third line of your immune system involves a very different set of cells that make up what's called the specific immunity, or the specific immune response, that is. And these can be thought of in addition. Move this stuff around here. Just go ahead and delete these. These are often referred to, kind of like we have two names for this. The other way to think about these is part of the adaptive immunity. Now, the way this works is you have cells called B and T lymphocytes. And the term lymphocyte is just a fancy term for cells. So these are more commonly referred to as B and T cells. Same thing as a lymphocyte, just that's the more technical name for it, lymphocyte. So these are both part of the specific immune response. And the way that these work is that you have billions, literally billions upon billions of specific types of B and T cells, and each one will respond to a particular what's called antigen. So in this particular case, rather than responding on a self versus non-self, so everything's either part of the body or it's foreign, in this case each cell will recognize one particular type of foreign particle or foreign microbe, and the term is called an antigen. We'll talk about this. By the way, this is actually the topic of chapter 15. So let me make a little side note here. This here is really what we're going to get into more detail when it comes to chapter 15. So I'm going to kind of keep this part of it brief here because we'll come back and spend more time on that now. So I'm just kind of giving you the overview, and so that we can kind of make more sense of it all. So this right here really is part of 14. Actually, kind of both of these here are part of 14. So B and T lymphocytes, each one will recognize a particular type of foreign entity and will respond to it on an individual case-by-case basis. So if you think about it, the second line is kind of like a general task force that's looking for anything considered quote-unquote bad. Almost like your kind of police force in the neighborhood or your local police force. They're just kind of there to take care of any sort of threat, whether it's a domestic threat or whether it's a robbery or whether it's could be a variety of different things. They're speeding, etc. Any kind of general threat they'd be there to take care of. The specific immune response is almost like the FBI or a more advanced level of a crime force agency. And what they're doing is they're looking at things on a case-by-case basis, and they're going after person X or person Y, and they're not necessarily just out there policing on a general basis. They're actually looking at a list of individuals who've done things specifically, and they're each part of that task force is looking at individuals on a case-by-case basis. That's kind of like how the third line of the immune system works. Each cell is delegated to one particular task, and as an overall whole, each cell is kind of taking care of individual foreign threats, and they all kind of work collectively as a whole. The other part of this are what are called antibodies. Antibodies are little protein molecules that are actually produced specifically by the B lymphocytes themselves. So these are the two cell types involved, and the B cells produce these acellular molecules called antibodies. And antibodies often have kind of a Y shape to them, and they tend to have they have an ability to attack pathogens and foreign particles without any connection to a cell. So B and T lymphocytes either actually like a cellular force, and these are just basically a protein that can attack molecules independently of other cells. So again, hopefully this will all make sense when we get to chapter 15. So anyway, that's your three levels. I wanted to give you an overview here of what these look like. So it starts with a barrier to infection, try and keep things out. It gets into a non-specific response where the body's kind of responding in very general terms, self versus non-self. And then anything that makes it past that will ultimately get into the third line where we have specific cells that are going to trigger specific types of responses. Okay, so let's take a look at the PowerPoint now. This is the first slide here after the title page. So here I'm referring to it as innate immunity. So we're starting with the second line of defense, and this is one more reminder. The first part is going to be discussed in the textbook, and it also touches on this as well, but we're going in more detail here. So once a microbe gets past the barriers to infection, the mucus, the skin, the different non-specific chemicals such as sebum and lysosome, all these things that the body has to kind of prevent microbes from getting in. If any microbe makes it past that, and eventually they will, then at some point they're going to run into different aspects of the second line. So these include things like phagocytes. Phagocytes exhibit phagocytosis, which is the action that they exhibit, whereas the phagocyte is the cell itself. So they'll run into phagocytes. They will trigger inflammatory events. Inflammation can lead to fever, and there's also a cascade series of, a variety of different what are called antimicrobial proteins. So these are kind of acellular immune components that will attack and try and ward off various invading microbes. So really, it's a whole series of reactions that all kind of work together. And I mentioned that it's kind of like a police force, and just like a police force, you have different parts of that. You have the sheriff versus the deputies versus the you can tell I don't really know my police force levels here, but anyway, you have different levels, kind of like you have at any agency, and this is kind of the same thing with your innate immunity. So one of the most common is phagocytes that exhibit phagocytosis. So these are typically one of the first components of the second line that foreign microbes and another, typically a living microbe like a bacteria or fungi, these are typically the first things that they're going to run into here. And we really have a variety of phagocytes. I've mentioned them so far as kind of one particular thing, like they all are the same, and the truth is they're not. There's actually a variety of them, and when we look at it in this chapter, we'll kind of break down those different variations. So the most common are what are called neutrophils, and then we have other cells called monocytes, and monocytes will actually develop into what are called macrophages, and then we'll actually see that we have different types of macrophages. So each one is capable of phagocytosis, the ingestion and swallowing and digestion of a foreign microbe, but each one is a little different, has their own characteristics. Now, as a whole, each one has really three general things that it's doing. And the first is surveying, and this is where the self versus non-self determination comes in. So they're surveying the body, and they're looking for anything that is determined to be non-self. And if it matches this kind of non-self description, then they will start to phagocytize it. Now, the immune system is very complicated, and we're really kind of talking about it in specific terms. If you notice here, it also says that they will look for particulate matter, injured or dead cells. So even in the case of the self, they can determine whether a cell is dead or otherwise basically on the way out, dying, injured, and they can actually determine that through different mechanisms and can be partly responsible for phagocytizing those as well. And that's a whole kind of a different part of the immune system. So we're really kind of focusing on the basics of it here in terms of how they survey microbes. So once they recognize a microbe as being foreign, then they will, and that actually gets into the recognition here. So really surveying, to clarify, involves kind of migrating throughout the tissue. Recognizing involves the self versus non-self determination. And then finally, the actual act of phagocytizing, which is ingestion and elimination of that particular microbe or particulate matter. Neutrophils, as I mentioned, are one of the most common, and neutrophils they make up something like 60 to 90% of all circulating white blood cells, just white blood cells in general. So they're very, very common. Really, if you think about the immune system as like a military force, then these are kind of like the basic infantry, and they tend to have the largest presence, and they also tend to have kind of a general role, not a great amount of specialization here. So typically a foreign microbe is going to run into these first in most cases. And so they're early in the inflammatory response to bacteria, foreign materials, and damaged tissue. Typically a sign of a bacterial infection is that you have a high neutrophil count in the blood. A much less common but also kind of general type of phagocyte are called eosinophils. And they tend to play a role when it comes to parasitic worm infections, as an example. So it says attracted to sites of parasitic infection, and this mostly talking about parasitic worms, although protozoa could also elicit the response. They tend to be attracted to certain types of infections like parasitic worms, and they often have a much more prominent role in that type of infection. They also play roles in what are called antigen antibody reactions, which is something that we'll talk about later when it comes to chapter 15 and chapter 16. We'll talk about disorders of the immune system. So these are both kind of very general types of phagocytes, neutrophils being the most common, and eosinophils being a type that would respond to certain types of infections, worm infections as an example of that. Okay, so the next cell type we're going to look at are the monocytes and macrophages. So Neutrophils and eosinophils are more of the kind of generalized phagocytes. Monocytes and macrophages are more of a specialized type of phagocyte. So you can think of them almost as like an elite type of white blood cell. Now, these two cells are connected in the sense that they actually begin as monocytes, and then monocytes will transform into macrophages. And so this takes place through various stages of the development, cell development. Cells start out as a stem cell. They ultimately will develop into a monocyte. Monocytes then are somewhat characterized by being found in the bloodstream. And monocytes then will become macrophages after they migrate out of the bloodstream and into the tissue. So you have stem cells in the bone marrow, which in this particular sequence will develop into monocytes, which travel through the blood, and then ultimately migrate into the tissue for their final stage of development. Now, really what's happening here is the body is creating monocytes in preparation for the need of macrophages. So what will happen is monocytes will circulate in the blood until they are stimulated. It says right here, due to chemical stimuli. So occurs, monocytes transform into macrophages after they migrate out of the bloodstream into the tissue due to chemical stimuli. And this has to do with other cells that are signaling them to come in to that area, which we'll talk about that later. So during a chemical stimulus, they migrate out and become macrophages. As I said, these are kind of like elite cells. You can almost think of a monocyte as like a cell in training to become an elite cell. And the macrophage is that cell at its kind of end of that elite training. So macrophages then are somewhat differentiated by the fact that they're no longer in the bloodstream, they're found in the tissue. They're larger in size, and they have an enhanced development of lysosomes, primarily two other organelles like peroxisomes, but lysosomes in particular. And if you recall from chapter 5, we talked about lysosomes being an organelle for digestion. So for example, I mentioned that these are kind of like the stomach of the cell, and they are specialized in digesting things. So in these cells, that capacity is somewhat tailored towards phagocytosis and breaking down harmful pathogens. Now, there are a variety of different types of macrophages. So there's some different terminology here that will reference different particular tissue types. So we have the monocytes, they become the macrophages. In many cases, we simply refer to them as macrophages. When they're found outside of these tissues down here and not in the bloodstream, we call them macrophages. Okay, so these examples would include the spleen, lymph nodes, bone marrow, kidney, bone, and brain. And so sometimes these macrophages travel back into the bone marrow after development. As I said here, they begin in the bone marrow. Sometimes they can travel backwards. So we refer to them simply as macrophages. Now, histiocytes are a specialized type of macrophage, and these are often characterized by where they're found, in addition to the fact that they often have some extra unique features. But examples include the alveolar macrophages. Alveolar macrophages are found in the lungs, alongside the alveoli, part of the lung tissue. So these are called alveolar macrophages. Kapuffer cells are found in the liver, named after Dr. Kapuffer, who named them after himself. And they're found in the liver. Dendritic cells are found in the skin around the dermal layer. So these are cells that have evolved to take on a special role in areas of the body that tend to see high amounts of pathogen or antigen come their way. So if you remember from chapter 13, we said the lungs are the most common portal of entry. So you have lots of microbes coming in there. So these cells are specialized to kind of play a role in that kind of high volume area, so to speak. The liver is a kind of a filtering organ. Lots of things come through there, and then the skin is susceptible to damage and all sorts of infection. So you have specialized cells there as well. So those are just some different terms that apply to different locations in the macrophages there. Now, Next thing I want to talk about here is a special role that macrophages play. Macrophages are a key cell that bridge the gap between the second and the third line of the immune system. So earlier I talked about the 3 levels of defense, the second level being the non-specific and the third level being the specific. Here's that diagram here. So we've got the second line and the third line. What macrophages are going to do is they're really going to trigger a connective kind of bridge, so to speak, between these 2 levels here. So these don't operate in a vacuum. They all tend to kind of overlap at times, even though we're kind of describing them as 3 different levels, or maybe think of them as layers. They're not totally separate from each other. So they definitely overlap and interact. That's part of what you read in the chapter 14 textbook questions. They're part of the textbook reading. So there's different ways that they can interact, and so we're not going to talk about all of those, but one of the common ways that the second line ultimately leads to a third line response is through the activity of macrophages. And really, it typically involves a cell called an antigen presenting cell, APC. But most commonly, these are macrophages. So there are other exceptions to the macrophage being an APC, but typically it's a macrophage. So we're kind of focusing on the most common scenario here. So what's going to happen is ultimately you're going to have some kind of microbe pass the barrier to infection. They're going to go from they're not going to be stopped here in the first layer. They're ultimately going to get into the second layer here. And that's going to trigger a response to a non-self entity. So the body is going to recognize there's a non-self entity there, and it's going to attack it. Now, what often will happen first is neutrophils, and for neutrophils, they will often be the first responder there. Neutrophils, going back here just real quick, neutrophils will send out chemical stimuli that trigger monocytes to come into the tissue and become macrophages. So neutrophils ultimately trigger the macrophages. Macrophages then act as an antigen presenting cell, and what they do is they start to gobble up some of the microbe. And when it comes to their roles in antigen presenting cell, they phagocytize, so they gobble up this foreign microbe, and then what they do is they break it down inside the cell, and they literally take parts of that microbe, little pieces of it, and they display it on the outer part of their cell. Then they take those many little pieces of the microbe, and they literally travel to the third line cells. In this case, they typically activate the T cells themselves, and the T cell activation then triggers that third line of defense to begin. So we go from a bypass of the barriers. Typically neutrophils are activated, triggers macrophages. They act as an antigen presenting cell. As an antigen presenting cell, they carry the antigen and they activate T cells. Now what happens next we'll talk about in chapter 15. T cells will activate B cells will create antibodies, and they all kind of act in concert at that point. But you can kind of see the connections here is what's happening. So barriers fail, non-self is recognized, typically a neutrophil leads to the activation of a macrophage. And by the way, there's more things here. I'm summarizing, I'm not talking every detail. There's not always neutrophils that have to activate the macrophages, but that's a common way. Macrophages phagocytize, acting as an antigen presenting cell, carry the antigen to the T cells and the B cells, and that activates your third line of the immune system. Now, just one more note about this here. Let's just stay here actually. These cell types here, the B and T lymphocytes, they tend to be found in two key areas of the body. Now, this is prior to their activation. So before they're activated, they tend to be found in the lymph nodes as well as the spleen. So these cells, before activation, are not just out patrolling the body like we see cells of the second line of the immune system. So these cells are really patrolling, scanning the tissue. If you remember just go back here real quick to remind you, some of the roles of phagocytes are surveillance. So they're out patrolling the body. It's not really true of the B and T cells. They're only activated, they're only in the body tissues after they've been activated. So prior to that, they're not actually scanning the body for the most part. So they have to actually be kind of woken up, so to speak. And so they're kind of sequestered away, and the way that they get activated or woken up is by the antigen presenting cell bringing the antigen, parts of the microbe, to the T cell. And then that triggers a variety of responses thereafter. So that's a key part of how the overall system works together. And the triggering of macrophages is the most common way that the third line is activated. And sometimes, to be fair, antigens can kind of bypass the first barrier and immediately trigger that third line. So, for example, something will just drain into the lymph nodes. So sometimes antigen from a microbe will simply trickle into the lymph nodes and activate these cells independent of a macrophage. So there's a variety of ways the immune system works, but most commonly it goes through these layers here, from barriers to neutrophils to macrophages to T cells to the third line being activated. Okay, so next I want to talk about some of the more specific events in phagocytosis. So we've talked about what that is, and now we're just going to talk about it in a little more detail here. So first of all, microbes are activated through what's called chemotaxis. And chemotaxis is defined as movement in response to chemical stimulus or stimuli, something to that effect. And this really applies in a variety of different contexts. Sorry, I'll leave that up there for 1 more second. Chemotaxis, movement in response to a chemical stimulus or stimuli. This is something that can take place in a variety of different contexts. So it's not specific to phagocytes, but in this context it can apply to phagocytes. So there's other ways that you can use that word in different contexts. But in this context, what it's referencing is the fact that your phagocytes are literally chemically attracted and move in response to stimuli given off by microbes. And you know what, I've got a really cool video I'll show you that will illustrate this better than I could ever explain. Okay, so this is off YouTube here. The title is called White Blood Cell Chases Bacteria. Fairly popular video as it looks here. And to point this out here, you have red blood cells here. These are red blood cells. This right here is a white blood cell. It doesn't say what kind it is, so I'm not exactly sure the exact type, but it is a white blood cell. And then right here, these two little black specks, these are a type of bacteria, appear to be a diplococci. Okay, so watch what happens next. So you can see that the white blood cell is literally chasing the bacteria. And this is taking place through chemotaxis. The white blood cell is almost like a dog sniffing out some kind of rabbit and chasing after it. Now it's you don't see all the details there, but what had happened there is it finally grabbed hold of it. Grabs a hold of it right about here and then quickly phagocytizes it. This is a very low resolution microscope, obviously, so we're not seeing all the details there, but that is a kind of the sped up process of phagocytosis. Now this, by the way, is a stop motion image, so it's being fast forwarded at a high speed, so it's not something that would take place this fast in reality. But in a kind of slow motion, you would see the same thing playing out in reality. So chemotaxis looks just like that. It's your body kind of chasing after, so to speak, these microbes. Wouldn't always have to do that, by the way. Sometimes it would grab them much faster. So chemical response there. Now the next step, I actually left it off the bullet point here, so let me point that out. The next step is actually adhesion of the bacteria. So it goes from a chemotactic response. Once they grab a hold of them, they adhere, and that involves the adhesion. So the bacteria have to stick to the cell. And if you remember back in chapter 4, I said that this is one of the problems with encapsulated bacteria, or bacteria with the capsule. It makes that process more difficult. They can't grab a hold of them. So normally speaking, they would grab a hold. You have these receptors here that grab a hold of them, and next thing is the cell swallows them, and that's the ingestion stage. So chemotaxis followed by adhesion, then engulfment, and the engulfment involves the membrane kind of sucking them into the cell. This then encloses them in what's called a phagosome. And a phagosome is basically a specialized vesicle that entraps these microbes. So this membrane invaginates backwards, seals itself off, and now creates a specialized type of vesicle called the phagosome. Now inside the phagosome, the bacteria are technically still alive. Nothing's really happened to them. They've only just been kind of sealed up. The next step is critical to their death, and that is that we have these lysosomes that we mentioned, key parts of macrophages, the digestive organelle of the cell. And the lysosome is literally going to fuse with the phagosome, and that involves then the formation of what's called the phagolysosome. So these two things fuse together, and this is kind of like dumping a bunch of toxic chemicals into the phagosome. So inside there with all these chemicals in, most of the microbes, if not all of the microbes, are going to be immediately killed, and this is a very effective effective concoction of toxic chemicals, and that will kill any microbes in there. So they're broken down, they're killed, they're degraded, chopped up in little pieces, so to speak, and in many cases they're excreted, which is the excretion of these bits and pieces now outside of the cell. So it's almost like a person, so to speak, or any animal eating, swallowing, digesting, and excreting. It's happening on an individual cell level. Now, one difference here would be if we're looking at an antigen presenting cell, one difference would be that rather than excreting everything, some of these components would actually be routed to the Golgi apparatus. If you remember from chapter 5, the Golgi apparatus is like the UPS of the cell. It's involved in packaging and shipping, and some of these pieces here would actually be packaged and shipped back to the membrane where they would be displayed for antigen presentation. So that part's not shown here, but that would be another kind of side event that would take place with macrophages. Okay, I'm going to post a link to this video online, or download, that is. You don't have to watch it, but it's just a nice little visual there of what's happening there. So back to the concept of chemotaxis. Chemotaxis is something that often takes place in response to what are called PAMPs, pathogen-associated molecular proteins. So if you think of the white blood cell as kind of like a dog, and the microbe as kind of like some kind of rabbit or squirrel, or maybe a raccoon if you're talking about a coon dog, then the PAMP is the particular smell or the particular characteristic that the dog will be picking up on, smell. And that's really kind of a way to think about it here. So, and this really kind of gets into an evolutionary context, because really what's happened over years of evolution is that our immune system has evolved to recognize things that are deemed to be foreign and harmful. And really what's happening is the immune system is profiling against particular types of recognized pathogens. So our immune system has evolved to key in on pathogen-associated molecular patterns, PAMPs. And so just to go back here and connect it to what I said earlier, this idea of non-self here, PAMPs are very distinct, recognized aspects of the non-self. So this is a complex thing. Non-self can take on many different contexts, and there's lots of different things you can get into here. But one of the most widely recognized concepts of quote-unquote non-self are these PAMPs. In other words, the body automatically recognizes them as being non-self. And that is, again, kind of based on a profile mechanism. So ultimately what they do is, there's something that the pathogen, that the phagocyte recognizes as being foreign. They serve as a signal molecule. And the key thing here is that these are not present in mammals where our immune system takes place. Obviously, we're a mammal, we're talking about the immune system here of a mammal. And so the key thing here is that these PAMPs have to be something that wouldn't be mistaken within our own body. So examples of PAMPs that you don't find in animals that would be exclusive to microbes would be peptidoglycan. Now think real quick, do you remember where peptidoglycan comes from? What about lipopolysaccharide? If you can't remember, these two things here both come from bacteria. Peptidoglycan is the primary component of a bacterial cell wall. That is where peptidoglycan comes from. So this is a cell wall component only found in bacteria. It's a molecule exclusive to bacteria. So by recognizing this molecule, the immune system can automatically identify anything with this chemical as being a bacteria, ultimately being of a bacterial nature. The other one, lipopolysaccharide, do it backwards this time, lipopolysaccharide. This exclusively comes from a gram-negative, this way, outer membrane. So the outer membrane of gram-negatives, as we said, the outer membrane is a key part of gram-negative bacteria. And this outer membrane is made of a certain chemical. This is just part of the outer membrane, it's not the entire thing, but there's a key chemical in there called lipopolysaccharide. And that chemical is only found in gram-negatives. So when the immune system recognizes either of these two chemicals, it automatically can associate them as being foreign, not being part of our body, therefore being non-self, and it's an immediate trigger that kicks off that phagocytic process. Another example is double-stranded RNA. We didn't talk too much about the different types of viruses back in chapter 6, but I did mention that there are RNA viruses, and I'll go ahead and clarify here. RNA is always single-stranded, with the exception of double-stranded RNA viruses. So these are literally the only time in nature that you will find RNA in a double-stranded form. Now DNA is double-stranded, but RNA is always single-stranded. Now we haven't talked about that yet. That's part of chapter 9, so I don't expect you to know that, or I'm not really going to ask you about that for any quiz or anything. But other than maybe I might ask you as an example of a PAMP, but I don't expect you to know the innate details of RNA at this point. But just to clarify, RNA is always in a single-stranded form, and DNA is always in a double-stranded form. Real quick, just to give you a visual, here's the side-by-side comparison. RNA is a single strand, DNA are two strands joined together. So the only time you would find a double strand of RNA, as I mentioned, is with a type of virus. So if the body sees that and it knows that's a virus. These are a few other examples, just to go show you a couple of visuals here and show you kind of the range of these. I pulled this off the web here. You have different types of PAMPs found in a wide variety of different microbes. So viruses can give off several different types of PAMPs. Gram-positive bacteria give off several different types of PAMPs. So all these here, like GP, DNA, RNA, DNA here, all of these are examples of PAMPs. Now they're all in acronym form, and this is a different figure for a different textbook. So I'm not going to use this for on the test or anything, but just to show you some of the different examples here. These can all be recognized as a PAMP here. Now the way the body actually recognizes the PAMP are through what are called pattern recognition receptors. So this kind of goes back to the lock and key mechanism that we find in many examples in biology. So I talked about it in enzymes and substrates, I talked about it with viruses and host cells, and once again we see it here. So the PAMP is like the key, and the pattern recognition receptor is like the lock. And in this case, the PAMP has to bind to a pattern recognition receptor. Now these are a little more general, not quite as specific as some of the lock and key mechanisms between viruses and host cells. They're more like a general key. It's almost like if you work at a building and they give you a key to the entire 5th floor, for example. That's kind of how these work. They're not quite as specific as other types of lock and key interactions, but they're still that same general kind of mechanism here. So these pattern recognition receptors are often found on phagocytic cells, dendritic cells, especially a type of macrophage, and then even some of our endothelial cells will have these as well. And that gets into a more in-depth part of the immune system that we really don't cover here. So, but these are basically the way to actually bind to the PAMP. And if we just go back here real quick, you see here that that's what's happening in immediate response to chemotaxis. So the chemotactic response allows the microbe or the phagocyte to adhere to the bacteria, and they're doing that with the pattern recognition receptor on the host cell. So that's actually how they lock onto them there. An example is of a specific type is called a toll-like receptor. These are the most common type of pattern recognition receptor. And so they are what specifically grab a hold of these microbes here. And if you just go back and look at this, TLRs here was showing how these different toll-like receptors bind to these various different PAMPs here. Okay, next we're going to talk about the inflammatory response. Okay, next we're going to take a look at the inflammatory response. And just to back up here real quick, inflammation is another part of the second line of defense, the innate or non-specific immunity. So really inflammation, and then we're going to talk about fever as well on these next 2 parts here. Now inflammation is a very broad topic, and it gets into a lot of different things. It's described in our textbook anyway as at least a response to a reaction to any traumatic event in the tissues. And so inflammation is something that we often just think of as swelling, redness, maybe pain, and that usually can happen due to a variety of reasons. So I should say it can typically happen due to a variety of reasons. So for example, a bruise is kind of like a type of inflammation. So if you hit yourself or something hits you or you fall down, it could happen if you get an infection. Maybe you have an infected hair follicle. Maybe you have an injury to yourself. You poke yourself with something. Maybe you burn yourself, or you have a chemical burn. Rashes. All these things are a type of inflammation, and so really it manifests in lots of different ways. So it's a very broad topic. So what we're going to do is we're going to look at it from a very specific type of angle. We're going to look at it from what we call an acute angle, or what's called acute inflammation, that is, rather. So inflammation, you can have acute inflammation, you can have chronic inflammation. Inflammation can be local, meaning it's refined or restricted rather to a certain area, or it can be systemic, traveling throughout the entire body or occurring throughout the entire body. And all of these can really be discussed in a different context. So a chronic inflammation is really quite different from acute inflammation, much less obvious. Systemic inflammation is going to take on different characteristics than local inflammation. So rather than try and break down the whole thing, we're going to look at it from an acute local inflammation standpoint. So in that sense, inflammation often is more specifically characterized by redness, warmth, swelling, and pain. And all these things can also take place on a systemic level, even on a chronic level, but not always to the same degree. And it's not always this extreme here, like you see in this picture, but oftentimes it's some degree of this. Now all of these really are characterized or are caused by increased blood flow to the area. And really that's what inflammation is all about. So redness, the color, is actually given off by increased concentrations of red blood cells. Same as the warmth. You have more blood flowing to the area. Blood carries warmth, and so the tissue gets slightly warmer. Swelling is caused by the fluid itself. Fluid is literally leaking out of the tissue or leaking out of the blood vessels rather into the tissue. And pain can sometimes be caused by the pressure of the excess fluid pushing on nerves in the area. And occasionally, if that happens to an extreme degree, it can cause decreased function. Like if you, for example, hit your thumb with a hammer, and then your thumb can't bend anymore because it's too swollen, although that's not as common. So these are all really a characteristic that results from increased blood flow to the area. And so as I mentioned, it can happen for a variety of reasons. Trauma from infection, injury, even one I didn't mention would be a certain type of immune reaction. And we'll talk about this in chapter 16 actually, but an example is like a type 3 hypersensitivity reaction. So these are a variety of different things that can cause it, some more common here. So we're really going to look at it in the next slide here, next couple slides, in response to really kind of an injury and infection simultaneously. Now, before we look at that, the functions of inflammation really serve kind of three general roles. And again, this is more of an acute local inflammation, really more of from an acute standpoint in general, I should say. And what's happening is the body is really signaling an alarm to some trauma in the body. And that alarm is ultimately geared towards attracting and mobilizing immune components. These include white blood cells, first and foremost. And the different signals that they give off bring in even more aspects of the immune system, like antibodies and things like that. So ultimately, it's an alarm system saying there's something happening here, we need to get some help over here, and so that is part of that process. I'll put these a little bit out of order. Once the immune cells get there, white blood cells, they're ultimately trying to destroy any microbes and prevent further invasion throughout the body. So they're not only going to destroy microbes, but they're going to kind of set in motion this kind of process that's going to wall them off and prevent them from spreading, sometimes referred to as granulomas. And then ultimately, they're going to set in motion repair mechanisms that allow the tissue itself to recover and reform, regenerate, and ultimately try and put itself back into as close to form as it was prior. So it looks something like this. And so this is again an example of where you have really an injury followed by infection at the same time. So this is some kind of needle or some kind of nail, perhaps. So it's piercing the tissue. So here's your skin, the nail or whatever this is piercing the tissue. And when that first happens, you've got injury, and the body is going to respond in kind of a knee-jerk fashion. And what's happening here is when this piercing event takes place, cells that line the tissue, these cells here are called mast cells, they're going to immediately release chemicals that are going to cause a response in that area. So it's very much like a knee-jerk reaction. And one of the first things that's going to happen is that the vessels in that area are going to constrict. The blood vessels are going to constrict, and we call that vasoconstriction. So it's a narrowing of the blood vessels. What that's going to do is it's going to kind of cause the tissue to kind of prevent immediate leakage. So in the very beginning, it's trying to kind of prevent fluid loss, and then that's actually going to change. It's actually then going to increase fluid loss shortly thereafter. The idea is that this kind of helps some of the clotting factors initiate, and it helps the body kind of start that repair mechanism by preventing excess blood loss. So you want to allow the wound to clot, and then the tissue will actually start to swell thereafter, which will then allow some of the cells, the immune cells, to come in. So the constriction kind of helps prevent excess blood loss. And so that's what you see here in this next stage. After the constriction has taken place long enough, and theoretically a blood clot will form that prevents excess fluid from being lost, excess blood from being lost, that is. And the next thing that's going to happen is now the vessels are now going to dilate. So you go from a constriction to a dilation. With dilation, what's happening is now you're going to have fluid that's going to leak out into the tissue area. From this figure, it looks like all the fluid would come through this opening here, and some of it certainly would. But even in areas outside the immediate wound here, and this is going to get clotted to a certain extent, keep in mind, what's going to happen is that the vessels are actually going to start to leak. So here's a figure off the web showing what this might look like. Under normal circumstances, the cells that line the blood, that make up the blood vessels, they're relatively tight and they form a relatively waterproof layer, not waterproof, but a relatively watertight seal. But the vessels themselves are not exactly like a hose. They're not watertight. They do have an ability to leak, and that ability to leak can be exacerbated during vasodilation. So it looks like this. You've got the vessels that all connect the cells, that all connect the vessels are somewhat locked together, almost like puzzle pieces, right? And then when vasodilation occurs, those puzzle pieces kind of loosen up, and you get these kind of gaps in between the cells. And that allows some of the fluid to literally leak out into the surrounding area there. So ultimately then, that is a big part of how the fluid gets into the tissue. And then also, as we're seeing here, some of the white blood cells are actually going to crawl out through those holes and migrate into the area as well. And going back here just real quick to connect this, what we said earlier, this is part of how the monocytes differentiate into macrophages, and they're actually going to crawl out of the blood vessels and into that tissue. And you can see here, once again, they're showing these kind of connected areas here that they would expand, and those would actually be able to crawl out. So ultimately then, we go from a constriction for clotting to a dilation to fluid seepage and accumulation, as well as cell accumulation in the area. So now things like neutrophils and the macrophages in short time would start to seep in. I misspoke there. These would be mostly neutrophils migrating into the area first. Ultimately then, the neutrophils are going to start to kill off a lot of these microbes, and the accumulation of pus is going to start to take place. Now this is going to take place over a certain number of hours, probably even days. So as the neutrophils start to kill off these microbes, pus starts to form. And pus really is a combination of white blood cells and phagocyte debris. It's kind of like a battlefield. It's like the remains of a battlefield left behind. So ultimately here you've got the body kind of fighting off infection beneath the surface here. Also, the body is laying down some of this fibrous tissue here, and that's theoretically going to help prevent microbes from getting spread throughout the body. So it's kind of trying to trap them in that area, prevent them from migrating further. And then ultimately, the body should theoretically handle these microbes, should handle the infection, and that should lead to scar formation. And as you can see towards the end, macrophages will come in. They'll scoop up, phagocytize lots of these microbes, and then travel back to the lymph nodes in the spleen, where they will trigger a third line of the immune response. This will allow the body to recognize these same microbes later and provide a more advanced immune response involving antibodies. Or if the infection is still not cleared, if this is becoming a long-term infection that's not being handled properly, the macrophages will go trigger a T and B cell response that can migrate back into the tissue and ultimately cause a more advanced response. Usually in this kind of scenario that doesn't typically happen, but it can. But in a kind of an average scenario, you would have clotting followed by fluid accumulation, which leads to pus, and then ultimately a resolution. And that's kind of a general sense of what acute inflammation would look like, acute local inflammation. So again, ultimately, steady motion repair, bring white blood cells, destroy the microbes in steady motion repair mechanism. Now, part of the big thing that's happening here are vascular changes, and these are really some of the things that are key to inflammation. And when you have a puncturing event or a bruising event or a trauma event, you're going to stimulate nerves, and it's, as well as some of the tissue there in the blood vessels themselves, and those are going to release chemicals that are going to trigger some of the vascular changes themselves. So if we're looking back here, I showed some of these mast cells, for example. They release chemicals, and that actually is what leads to vasoconstriction, and then ultimately different chemicals lead to vasodilation. So any kind of chemical that affects the blood cells, whether in a constrictive or dilative way, these are considered vasoactive mediators. Other chemicals that are released have an effect on the white blood cells themselves. These are called chemotactic factors, sometimes called chemokines, and they're the chemicals that will actually draw in some of the white blood cells, like macrophages. Now, some of the terms here that relate to how macrophages and phagocytes are changing their behavior. One of them is called diapedesis, and then chemotaxis. These actually kind of go hand in hand, so we'll talk about both of them. Remember, chemotaxis we described as being a response in chemical movement, in response to chemicals. And so what this is again applying here. So the white blood cells are chemically attracted, in this case, mostly to chemicals given off at the site of the injury or infection. So they could be PAMPs, the pathogen-associated molecular patterns that we said earlier, but probably more prominently in this case, they are chemicals given off by your own cells as a response to a trauma. So, for example, some of the mast cells releasing chemicals that would ultimately attract them in. Neutrophils release chemicals that attract macrophages. So chemotaxis is kind of, in this case, a general term in which the white blood cells are migrating towards the traumatic event. And diapedesis is a term that describes how they literally crawl through the vessel lining here in your blood vessels. So the vessels, I mentioned, are kind of connected like puzzle pieces here. And as they dilate, these white blood cells can literally crawl through them, travel out of the vessels and into the tissue around them. And that's ultimately how they get from the vessels into the tissue. So that's called diapedesis. Okay, so that's a quick summary of acute inflammation there. Next, I'm going to talk about fever just shortly here. Fever, as we know, is an increase in body temperature. We've all probably experienced this at some point. Fever, though, is actually something that is controlled by a part of the brain called the hypothalamus. Hypothalamus does many things, so it's not just what controls fever. It's a very complex endocrine part of the endocrine system in our brain, and controls many different aspects of our hormonal system in the body. So a very complicated part of the brain. But one of the many things that it does is it plays a role in fever. And fever ultimately is a response to molecules called pyrogens. And a pyrogen is a fever-inducing molecule. The term pyro relates to fire and heat. So think pyro, think fire, think pyrogen, fever. And ultimately what they're doing is they're causing the hypothalamus to reset itself to a higher temperature. So you can really almost think of in the hypothalamus in this context as almost like a thermostat on the wall that controls the room temperature. And a pyrogen is basically the equivalent of someone pushing the button and increasing the temperature. And ultimately that causes the body temperature to rise. Now pyrogens can typically be of several different sources. Many of them will occur inside the body. These are called endogenous. They originate inside the body. Most often these are produced by phagocytes. So phagocytes are obviously the white blood cells that phagocyte, and anyway they produce chemicals during that phagocytic process. And there's a threshold here. They don't do it right off the bat. So this is a more in-depth part of the immune system, but there's kind of this delayed effect where if the phagocytes are active long enough and there's enough of them and they reach kind of a quorum, then they will eventually start to release pyrogens. So that's not really a knee-jerk thing, but more of a delayed, kind of long-term in response to a more traumatic type of infection. This is why you don't get a fever every time you have a little minor hair infection or like a hair follicle infection or something like that. So they're not just going to automatically trigger fever right off the bat. But that can ultimately take place due to phagocytes. Occasionally, you'll be exposed to chemicals, pyrogens, from an outside source. These are called exogenous, and these are the different kind of different class of pyrogens that can ultimately do the same thing, but they come from an outside source. So some examples of these would include some of the products of things like viruses, bacteria, protozoa, and even fungi. So some of these types of pathogens can actually give off chemicals that ultimately cause your body to go into a feverish state. One example is endotoxin. Endotoxin comes from the lipopolysaccharide I mentioned as being a type of PAMP. I mentioned that being part of the gram-negative outer membrane. So gram-negative bacteria are particularly known to induce fever given a relatively serious infection. Now, once again, they're not going to do it just automatically. There are kind of threshold levels here, but they are able to cause a fever if they have a serious enough infection. Some species are more potent at that. For example, Salmonella typhi is a microbe that causes typhoid fever. Typhoid fever is a name given to the fact that that particular infection is well known to cause a high fever. Other things like vaccines, injectable solutions, even certain types of blood products, like if you have an inappropriate blood transfusion, these things can ultimately cause a pyrogenic response. Endogenous, as I mentioned, often come from phagocytes like monocytes, neutrophils, and macrophages, and can ultimately trigger fever due to a kind of a natural immune response. That's most typically how that's going to take place. These are typically a natural response to infection. Now, one of the reasons why the body wants to do this in the first place, because fever, as you may be well aware, fever can be a real problem. If the fever gets too bad, then it could actually be detrimental, even fatal in some cases. Our body has kind of a sensitivity to too high of a temperature, obviously. And sometimes the fever can be very detrimental. But ultimately, the idea behind the fever is that it does have benefit at the right level. So if it's not too high, there is a benefit to it. And one of the benefits is that certain microbes are temperature sensitive. And if the temperature gets too hot, they just simply can't replicate, they can't function properly, and that can be an advantage to the body. It kind of either slows them down or prevents them from replicating. So certain types of cold viruses, like a rhinovirus, for example, is really not able to replicate at a high temperature. So if the body's fever goes up, the microbe replication goes down. When it comes to certain bacteria, bloodborne bacteria, like streptococci and staph, the ability for them to absorb iron goes down as body temperature goes up. So this is kind of an enzyme substrate complex interaction that is reduced due to a higher temperature. So a slight amount of denaturation of the enzyme occurs, and iron absorption goes down due to that denaturation. So it's a sensitive enzyme. So the idea is that iron is often a limited nutrient, and it kind of limits the rate of their growth. So microbes can only grow as fast as the least amount of nutrients that they have. So they may have lots of carbon, they have lots of hydrogen, but iron is necessary for growth, and it's usually the most difficult to get. So by slowing the rate of that particular nutrient down, it slows their overall rate of growth down as well. So that can help the body gain a foothold on those microbes. And ultimately, it's thought to increase the metabolism and stimulate kind of a general increase of immune response throughout the body. When things heat up to a certain degree, they tend to move faster. And so as the body heats up, theoretically things move a little faster, the immune system speeds up, and circulation speeds up, and things are kind of taking place at a slightly faster speed. Again, this all has thresholds, at about 101, maybe 102 degrees. There may be a lot of benefit to that. You get up to 105, anything past that, and suddenly you're looking at some pretty serious consequences. So we're all within reasonable limits here. So that's kind of the benefit of fever and kind of how fever works here. The last little bit here is taking a look at a couple examples, go all the way back here, of antimicrobial proteins. So we've talked about phagocytosis, inflammation, fever, and one of the last examples of innate immunity are antimicrobial proteins. So these two that topic in general, antimicrobial proteins, can really get complicated. So I'm going to really try and keep this simple here and not go into a lot of detail. So if you look at your study guide, just have a couple questions about the two examples I'm using here. So nothing really in depth. So I'll try and keep this last little bit short here. But interferon is a really interesting process that takes place in response to viruses, typically. And when a host cell rather is infected by a virus, one of the things that our cells have evolved to do is to literally send out a signal to other cells in the nearby area that they have been infected, or that cell has been infected. And what happens here is the cell releases a chemical that will bind to the nearby host cells, and that activates internal cell pathways that are typically effective at breaking down viral DNA. So when a DNA virus comes into the body, it will typically infect a certain number of cells, depending on how many virus particles you're exposed to. And each of the initial cells that are infected will put out an interferon signal, which will then trigger other cells in the neighborhood, nearby tissue, that will give off that will activate a pathway that will prevent them from being infected at the same rate. So it's a lot like a warning signal. It's almost like if you have, in a neighborhood, if you have someone going around breaking into houses or breaking into cars, one house gets broken into, maybe two houses get broken into, and they're going to signal to the rest of the neighborhood, hey, we've got someone going around breaking into houses here. And everybody in that neighborhood is now going to be on alert, right? Especially in Kansas, where everybody's got a gun. That criminal is probably not going to break into too many more houses successfully. So it's kind of like that. You've got a small number of incidences that lead to a warning to everybody else to be on alert, and that makes it much more difficult for, in this case, the virus to be effective at continuing to infect. So it looks kind of like this. The virus comes in, this first cell is infected, but what it does is that triggers a synthesis of this interferon chemical. And that interferon is released and binds to other cells in the neighbor area, and that changes their response and ultimately activates pathways that otherwise wouldn't be active. And those pathways ultimately will break down incoming viruses as they attempt to go on and infect those other cells. Once these pathways are active and the virus tries to infect them, it breaks down the virus after it comes inside the cell. So this is kind of an elaborate evolutionary mechanism to viral infection. And this is one of the reasons why our body is less susceptible, I should say, to massive infection by virus. If you mentioned from chapter 6, we're talking about viruses. One virus can potentially make hundreds of thousands, if not millions, of copies of itself in just one cell. And then each one of those could theoretically do it again. So if you do the math, one virus could easily lead to a million more viruses, and each one of those viruses could lead to a million more, and so the numbers would be overwhelming relatively quick. One of the things that stops that is the fact that our cells put out signals that often prevent them from continually infecting every cell thereafter. Here's another video. I'll post a download for that online also. And the last example here is another type of antimicrobial protein. These are called complement proteins. Complement proteins work in conjunction with the third line of the immune system. So they actually work in conjunction with antibodies, which are produced by the third line of the immune system. So these typically take place as kind of a combination between the specific and the non-specific. So they're technically part of the non-specific. And the reason for this is because they will typically bind to any type of antibody on the surface of a cell. So they don't necessarily have to recognize a certain type of antibody, so kind of any general antibody. So the antibodies are specific, but the complement proteins are not. They just pretty much recognize any kind of antibody. And what happens here is antibodies will tag or coat a cell, like a bacteria. And so that kind of marks these cells, and that indicates that they need to be eliminated. So these complement proteins will then recognize those, and they will bind to the antibodies. And what happens is that the binding of these complement proteins ultimately sets off a cascade of reactions. One protein binds, another protein binds to it, and it follows in suit in a cascade reaction. And the binding of all these different antibodies ultimately triggers a perforation in the cell membrane of the microbe. And so what it ultimately leads to are these puncture sites in the hole in this cell of a microbe. So this is an interesting figure here. It's actually showing the surface of a microbe, and each one of these little holes, like a bunch of craters in the surface, each one of these are actually a perforation by these complement proteins. So the antibodies tag the microbe, and then these complement proteins come in and literally start to align themselves in a way that punctures holes in the cell. Now the chemistry behind this is actually fairly complex, and this is one of those areas I just kind of choose to not make you learn and get into all the details of that, because it's something that you can spend quite a bit of time actually learning exactly how this all works. And there's all these complicated mechanisms by which the first protein and then the second, third, and fourth proteins all bind. It's a little tedious. So rather than get into all that detail, we'll kind of talk about the general process. So it's a protein in the blood, a protein series of them, that bind to antibodies. And when they bind, they ultimately will lead to an arrangement that perforates a hole in the cell. So it's almost like this little pattern that aligns itself on the surface of the membrane, and then there's a process that literally punctures a hole after these proteins line up in just the right mechanism. Pretty interesting stuff, actually. But ultimately, the end thing here is that it punctures holes in the membrane, and that causes lysis, popping or rupturing of the cell, pathogen, and death. So this takes place after you've been exposed to a microbe, but it's considered non-specific because these complement proteins will typically bind to just any kind of antibody here that has tagged a microbe. Okay, so that is the end of chapter 14. Make sure you take a look at the textbook questions if you haven't done that already. As always, let me know if you have any questions, and I will get back to you as soon as possible.